At 4,500 meters above sea level, the Tibetan Plateau should be uninhabitable for humans. Most people would suffocate. Their blood would thicken dangerously within hours. Yet Tibetans live here with ease, defying everything we thought we knew about evolution. The real secret behind their survival was not found in living bodies, but inside a finger bone that is 50,000 years old from a Siberian cave. How could ancient DNA solve a high-altitude mystery that stumped science for decades? The Tibetan Plateau stretches for thousands of kilometers, rising to heights where the air holds just 60% of the oxygen found at sea level. At these elevations, the human body faces a relentless struggle. Within hours, most people develop pounding headaches, nausea, and confusion as their brains and muscles starve for oxygen. The medical term is hypoxia, a condition that can quickly spiral into high-altitude pulmonary edema, where fluid fills the lungs, or cerebral edema, which can cause coma and death. The body tries to compensate by pumping out more red blood cells, thickening the blood in an effort to capture every molecule of oxygen. But this response comes at a steep price. Blood that is too thick strains the heart, increases blood pressure, and raises the risk of stroke or heart attack. Even healthy adults, after only a short stay at these heights, may find themselves gasping for air, their hearts racing, their judgment clouded. For most of human history, such high-altitude environments have been nearly impossible to conquer. Expeditions to the plateau's remote valleys and wind-scoured ridges often ended with teams forced to retreat, overwhelmed by the invisible pressure of thin air. The plateau's climate compounds these challenges. Temperatures can plunge below freezing even in summer, and winter storms sweep across the open grasslands without warning. Yet, against all odds, entire communities have not only survived, but thrived here for generations. Their livestock graze at elevations that would leave most animals breathless. Children play and run with no sign of fatigue. For decades, scientists searched for answers in diet, training, and culture, but none could explain how Tibetans managed to avoid the fatal consequences of hypoxia. The speed and scale of their adaptation defied the slow march of natural selection. Something in their biology was rewriting the rules of human survival at altitude, hinting at a solution far older than their own history on the plateau. In 2010, inside a secure laboratory at the Max Planck Institute for Evolutionary Anthropology, a team of scientists hovered over a tiny ancient fingerbone. The fragment came from Denisova Cave in the frigid Altai Mountains of Siberia. At first glance, it seemed unremarkable. Just a sliver of the little finger from a young girl who lived nearly 50,000 years ago. But as Svante Pääbo and his colleagues extracted DNA from the bone, the results defied expectations. The genetic code did not match modern humans, nor did it fit the Neanderthal profile. Instead, the sequences revealed an entirely new branch of the human family tree, a lineage distinct from anything seen before. The researchers named this enigmatic population Denisovans, after the cave that had sheltered their remains for tens of thousands of years. The discovery electrified the scientific world. Until that moment, the story of human evolution in Eurasia had centered on two main characters, Homo sapiens and Neanderthals. Now a third player had entered the scene, one whose existence was known not through a trove of fossils, but through molecular signatures preserved in ancient bone. The Denisovan genome carried secrets that would soon reshape our understanding of human ancestry. When the team compared this ancient DNA to genetic data from people across Asia and Oceania, they found traces of Denisovan heritage scattered through modern populations. In some Pacific Island communities, Denisovan ancestry accounted for as much as 6% of the genome. Even people in East and Southeast Asia carried small but detectable fragments of this archaic DNA, inherited from encounters tens of thousands of years in the past. The presence of Denisovan genes in living people raised urgent questions about ancient migrations, interbreeding, and adaptation. How did these mysterious hominins interact with our ancestors? What legacy did they leave? As researchers pored over the data, one thing became clear. The genetic influence of Denisovans extended far beyond the cold caves of Siberia and into the biology of people living today. The search for answers would soon lead scientists to the windswept heights of the Tibetan Plateau and to a genetic adaptation that defied the ordinary pace of evolution. Rasmus Nielsen, a geneticist at the University of California, Berkeley, set out to answer a question that had stumped scientists for decades: what allowed Tibetans to thrive where others could barely survive? In 2014, Nielsen and his team compared the genomes of 40 native Tibetans with 40 Han Chinese, searching for genetic clues buried in their DNA. The results pointed to a single gene, EPAS1. This gene acts as a master regulator for how the body responds to low oxygen. It controls the production of hemoglobin, the molecule that carries oxygen in red blood cells. In most people, high altitude triggers a surge in hemoglobin, thickening the blood to dangerous levels. But in Tibetans, the EPAS1 gene works differently. Their version keeps hemoglobin levels in check, preventing the blood from becoming too thick and reducing the risk of heart attack or stroke. What made the Tibetan variant so remarkable was how distinct it was from versions found in any other human population. Nearly 87% of Tibetans carried this unique sequence, while it was almost absent in Han Chinese and other lowland groups. The variant allowed for efficient oxygen delivery without the deadly side effects seen in unadapted populations. Nielsen's team found that this genetic difference explained why Tibetan children could run and play at high altitude without the headaches, fatigue, or heart strain that plagued visitors from the lowlands. The discovery of EPAS1 provided the first concrete evidence that a single gene could dramatically alter how humans cope with extreme environments. But the mystery deepened. How had such a powerful adaptation appeared so quickly in a population that had only lived on the plateau for a few thousand years? The answer, it seemed, would lie not in modern DNA, but in the genetic legacy of an ancient and long-lost branch of humanity. In 1980, deep in the shadowed mouth of Baishia Karst Cave, a Buddhist monk brushed away loose earth and uncovered a heavy jawbone. The fossil, thick and weathered, traveled from the monastery to a university drawer, where it would sit for decades. Only with the arrival of advanced protein analysis did its true story come to light. In 2019, scientists extracted collagen from the bone and compared its sequence to every known branch of the human family tree. The result was unmistakable. This mandible belonged to a Denisovan who lived at least 160,000 years ago, high on the Tibetan Plateau. The existence of Denisovans at such altitude, so long before modern humans arrived, offered a crucial clue. Here was proof that an archaic human species had survived, hunted, and thrived where oxygen is scarce and winter never truly ends. But the real breakthrough came when geneticists traced a remarkable thread running from these ancient cave dwellers to the present day. Through a process scientists call adaptive introgression, a handful of Denisovan genes, carried into the gene pool by interbreeding tens of thousands of years ago, swept rapidly through populations settling the plateau. Among these, the EPAS1 variant stood out, enabling efficient oxygen use without the deadly side effects of thickened blood. By 2025, new genomic surveys of Himalayan peoples confirmed not only the Denisovan origin of EPAS1, but also revealed additional archaic genes, such as variants in TBC1D1 and PRKAG2, fine-tuning high-altitude adaptation. Each discovery tightened the causal link, showing how a single fossil, found by chance, unlocked the story of how Denisovan DNA continues to shape the survival of millions on the roof of the world. Today, the genes of an extinct species help millions thrive, in places where survival once seemed impossible. As we unlock more ancient DNA, our own story grows less separate and more intertwined. Human evolution is not just history, it is an ongoing inheritance. What else waits in our blood, still shaping us now?